Hello, everyone, and happy holidays. Thanks for joining us. I'm Dave Iverson, the Michael J. Fox Foundation's contributing editor and the moderator for today's Third Thursday panel, which is, of course, is part of our continuing series of Third Thursday webinars where we ask you to join us on the third Thursday of every month at 12 Eastern, 11 Central, 10 Mountain, and 9 Pacific to take up a topic of interest to the Parkinson's community. And we hope you'll continue to do that as this series continues throughout 2016. As always, you'll be able to submit questions throughout the hour, which our experts will be able to take up. In the second half of our program, there's a Q&A box that you'll see near the middle of your screen, and you can just type your questions in, and we'll do the best we can to get to as many of those questions as possible. As always, of course, though, we won't get to everything, and so we have, as usual, our extra opportunity to pose questions, which is on Twitter. That'll be at 12 noon Eastern tomorrow using the hashtag FoxPDChat, where we'll continue our conversation about the state of research in 2015. A couple of other quick last notes. We're going to provide our resources to you on the resource list. You'll see that box. You can just click on that link, and the document will open in the new browser window, and you can save or print that up from there. And then finally, we also want to make sure we thank our Third Thursday webinar sponsors who have sponsored us throughout the year: Acadia Pharmaceuticals, Medtronic, and Teva. All right, let's get started with what we're going to talk about today, which is the state of research as we come to the year's end. We're going to be talking about what the state of therapies is to slow down or stop Parkinson's disease. We'll talk some about new treatments for the motor symptoms of Parkinson's as well as the non-motor. Spend some time talking about new technology, connecting patients and researchers, and then we'll also talk about what people can do today. Our goal really overall is to just give a snapshot of where we are in research, where we've come, and where we hope to go in the year ahead. Let's meet who's going to be joining us for our panel today. We're joined by Dan Cannell. Dan is a law partner in Rochester, New York. He was diagnosed with Parkinson's disease in his early 40s, and as you can tell, he spent part of the year doing safari research. It doesn't have a lot to do with Parkinson's, but Dan is actually the king of participating in clinical trials, and Dan, we'll hear more from you later in the program. Thanks very much for being part of this conversation.
Thank you.
Joining us as well is Dr. Tim Greenemeyer. Dr. Greenemeyer is the director of the Pittsburgh Institute for Neurodegenerative Diseases and the chief of the Movement Disorders Division at the University of Pittsburgh's Medical Center. Dr. Greenemeyer, thanks for being with us.
My pleasure.
And joining us as well is Mark Frazier. Dr. Frazier is the Senior Vice President of Research Programs at the Michael J. Fox Foundation. Mark, thanks for being with us as well.
You bet. Glad to be here.
All right, let's get started with where we are with our topic. We've got a big list on the screen now that's titled Promising Therapies that May Slow Disease that are now in clinical trials. And Mark Frazier, I think one of the great things about this list is that it's not a wish list. It's kind of a work in progress list. What's so striking, I think, is to think that no longer are we kind of dreaming about the possibility of finding a way to stop or slow Parkinson's disease. We've actually got a half a dozen or more ways to do that are now being tested. I think it says a lot, don't you think, about how far we've come?
Yeah, I think that's right. And for a number of years, there were several different hypotheses about how one may slow the progression of Parkinson's disease. And they were tested in a petri dish or test tubes, but this slide really illustrates that a lot of the hypotheses are being tested in humans, and we'll learn a lot from these trials. Some of them may succeed, some of them may fail, but we will certainly learn about Parkinson's disease and what may slow the progression of Parkinson's through these variety of hypotheses that are being tested in these trials.
So let's kind of group them if we can and give people a sense of where we are with making progress towards that goal. We see, Tim Greenemeyer, the word alpha-synuclein in each of the first three strategies that are mentioned. That, of course, is the sticky protein that clumps together in Parkinson's and we think is responsible for cell death in Parkinson's disease. And the first three of these strategies are about ways to clear out alpha-synuclein. Describe a little bit more, if you would, Tim Greenemeyer, about what that strategy is and what we've learned so far as these clinical trials are ongoing.
Sure. I think I'll back up a little bit. And as you said, alpha-synuclein is an important protein in Parkinson's disease. We know that if people have mutations in the gene that makes alpha-synuclein, they get the disease. But if you simply make too much of it, you'll also get the disease. And most people have heard of the Lewy bodies that accumulate in Parkinson's disease. And alpha-synuclein is the main component of those Lewy bodies, even if you don't have mutations or make abnormal amounts of it. So it's clearly an important player in the disease, and we know that it causes toxicity to the cells that ultimately get sick and die in the disease. So the first three strategies are basically ways to try to reduce the levels of alpha-synuclein in the brain and see if that will reduce the toxicity, perhaps even rejuvenate some of the sick cells if they can unload some of their alpha-synuclein. And the ultimate goal then is to either slow down or, if we really hit a home run, to stop the disease in its tracks.
And Tim Greenemeyer, on the one where it says, what's the difference? It sounds like a nuanced difference, but I'm sure it's an important one between the first three, which say clear alpha-synuclein, and then Neuropore, which is the drug designed to prevent the clumps, I guess, from actually pouring. Is that an important distinction? And describe, if it is, what that distinction is.
Yeah, the first three strategies will reduce overall levels of alpha-synuclein, and I think that's a reasonable strategy. The fourth one, the Neuropore, is a little bit more targeted. As alpha-synuclein begins to form its clumps, it goes into a different state. Small amounts of it aggregate together or clump together. And this is really trying to prevent that initial clumping of alpha-synuclein into the Lewy body. So it's going after sort of specific forms of alpha-synuclein rather than overall levels. So it's a little bit more targeted approach, and I think it's a little bit higher risk potentially, but we'll see as the trials proceed.
And by higher risk, Tim, what is the risk? Is it to patients or is it?
Oh, I don't mean. I'm sorry. I think it's a less of a sure shot of working. I didn't mean more of a risk to patients. No.
Okay, and Mark Fraser, we see at the top, the graph that tells us how far along we are, and the first couple are kind of entering phase 2. Can you just give us a brief layman's description of what that means and how far it will be till we get to the end of phase 3, which is when we'll really know whether or not one of these strategies work?
Sure, and this is one of the unfortunate things about neuroscience research, and especially Parkinson's research and drug development, in that it does take a long time to get through these different stages. And it's, of course, an expensive process. But very briefly, phase 1 is the first time that a drug has been tested in humans, and really the goal of those studies are really just to determine whether the drug is safe and tolerable, can be tolerated in human beings. Oftentimes, those trials are done in a healthy population, so not in Parkinson's patients. And those are shorter duration trials over the course of several weeks, sometimes months, depending on the approach. But it's really in the phase 2 process where the trials are tested in the patient population, in this case in Parkinson's disease. They're a little bit longer in duration. The overall goal is still primarily to understand whether it's safe and tolerated over a longer period of time, but it is in Parkinson's patients. And we can get an initial read and some data on whether we think it may be modulating the symptoms or the progression of Parkinson's. And then it's really, and so those studies may be 9 to 12 to sometimes 15 months in duration. But in phase 3, particularly around disease-modifying trials where the goal is really to demonstrate in a large number of people that the drug can change the course, the progression of the disease. These require a large patient population and a long period of time really just to follow and track individual symptoms to see if the drug is making a difference on the progression of the disease. These can be 18 months to 24 months, sometimes even longer. And so that is the process to really generate a large amount of data, one, to make sure that these drugs are safe and tolerated in a patient population, but then ultimately to generate data to go to the FDA and demonstrate that the drug is actually making a difference. So that's generally the timeline of these different phases.
So still several years out before we'll know for sure which of these will actually play out and hopefully be successful. But I think that, again, the key thing is that there are so many possibilities. Let's move to the next one, which has gotten a lot of attention recently, which is nilotinib, which is actually a cancer drug. And Tim Greenemeyer, it's kind of a hard concept to get your head around, that a cancer drug would somehow have potential to be effective in slowing down Parkinson's disease. Can you describe what the drug is and how it might work?
Sure. And I would say that most of the data on this related to Parkinson's disease, just as a preface, is cells in a dish kinds of experiments, but it looks promising in that regard. Basically what nilotinib does is increase a cellular process called autophagy. And you can think of autophagy as the garbage disposal or the system to get rid of cellular debris that might go awry in Parkinson's. And if you don't have a way to get rid of the cellular debris in the cells that are sick, they just accumulate that garbage, and that leads to accumulation of things like the alpha-synuclein and formation of the Lewy bodies. So this kind of stimulates that garbage disposal system and has been shown to, in cells in a dish, decrease the alpha-synuclein and the aggregated alpha-synuclein. So it looks promising in that regard. Regarding the human data, there's one very small trial from Washington, D.C., in a dozen patients. But I think all that we can draw from that trial is that it appears to be safe at the doses that were given. It was an unblinded study that involved only about a dozen patients. So although there was a great deal of press about it, I would say that the data that it actually works as we hope it will in Parkinson's disease is essentially zero at this point. But there are trials being planned where we should get that information.
And by unblinded, Tim, you're referring to the fact that there weren't any controls. And the reason why that's important is that if you don't have a control population, it's hard to know whether or not what you're reading is a real effect or whether or not it's a placebo effect. Is that right?
That's right. And all the patients knew that they were getting active drugs. And so what often happens, especially in Parkinson's disease trials for interesting reasons, is that there's often a big placebo response. So people think they're getting the active drug, or in this case, they know they're getting the active drug, and they get better just based on essentially the hope that they're going to. And that effect can be very big and very large, long-lasting.
It is indeed a fascinating phenomenon. You wish you could bottle the placebo effect.
Yeah, absolutely.
Before we get to our last two, let me bring Dan Cannell into our conversation, because one thing, Dan, that I think is striking again when you see all these options is, as a patient, how do you know which trial to participate in? There are so many out there. It's kind of a problem of riches, which is a good thing. But you're someone who has participated in 8 or 9 different clinical trials. And I'm curious about the process you go through when deciding which clinical trial to participate in.
Well, I think that prior to deciding what trial to participate in, it's important to note why it's important to participate in clinical trials. There's lots of researchers doing amazing work, thinking of all kinds of ideas on potential cures and symptomatic relief possibilities. But the only way to move that process forward is to have people to participate in trials and actually test the drugs and test the therapies. I read someplace that more than 80% of all trials are delayed because of lack of participation, and a sizable number of trials don't even get a single participant, that they're canceled because of a lack of participation. So I think that anybody interested in moving the ball forward and kind of enhancing the number of shots on goal to find a cure needs to be active in participating in clinical trials. As far as deciding which ones to participate in, I think there's a whole range of options in terms of what to participate in. I think the easiest way to sort of whittle down the number of trials that are available is to use the Fox Trial Finder, which is a great online resource and tool that you can put in some personal data about yourself, and it'll send you information about trials that you may or may not be interested in. And then you can contact the trial teams and see what's out there, see what's in the geographic area you're willing to travel to, and take a look and see if there's all kinds of options in terms of disease-modifying therapies. There are studies about gait and wearable devices. There's a wealth of options to help advance research, and it's not always about taking medication or getting something injected into you that you can participate in. And I think the key is to participate. The key is to keep the process moving forward and give the researchers the tools they need to help advance the science.
Thank you, Dan. And we'll put up how to get engaged in Fox Trial Finder a little bit later in our discussion today. Let's take up our last two items on this long work in progress list, and then we'll move on to what's going on with symptomatic treatment in Parkinson's. And Mark Fraser, back to you on these last two. Is Radipine, which is actually yet another drug for initially a different disease, this time for hypertension. Inosine is something that is a different drug, which is to boost uric acid levels, something that we hope might be neuroprotective. These look, Mark, like they're quite far along. So in this case, particularly with this Radipine, we'll know the answer pretty soon, right? Whether or not this blood pressure drug might actually work. Tell us what it is and how soon we'll know.
Yeah, this is a really interesting story. And again, going back to my comment earlier, it was based on a hypothesis that had been in the field for a while, and there were some experiments done in preclinical models to demonstrate that we think that by reducing calcium levels in dopamine cells, which are the cells that deteriorate in Parkinson's, that could provide benefit and potentially rescue the loss and the death of those cells. And so the Fox Foundation actually supported the initial phase 2 studies of this work, and the trial demonstrated that in addition to being safe and tolerated, this drug that is typically used for blood pressure, and it's been approved for reducing blood pressure for some time, this drug actually could have an effect on Parkinson's symptoms and slowing the progression of Parkinson's disease. And so the phase 3 trial just finished recruitment earlier this year, I think, and that trial will follow individuals. I believe it's over 2 years. So within 2 years, we'll have a signal of whether in a large study whether this blood pressure medication actually changes the progression of Parkinson's disease. So it's a really unique example of a drug that was approved for one indication completely independent of Parkinson's, but because it was approved and used in so many humans already, the path to the clinical testing was a lot quicker, and so we repositioned it to be tested in Parkinson's disease.
And last on inosine, Tim Greenemeyer, this is something that, as I mentioned, boosts levels of uric acid, which we think may be neuroprotective, in some way ward off some of the oxidative stress, as I understand it, that takes place in Parkinson's. Explain briefly the concept.
Yeah, you're absolutely right. Urate or uric acid, which are the same thing, is an antioxidant compound, so it reduces some of the oxidative stress and oxidative damage in cells. The idea for this study came from epidemiologic studies that found in studying very large populations that if you had high uric acid levels, like people with gout do, but even people without gout who have high uric acid levels, you had a lower chance of getting Parkinson's disease. And then people began to study this in cells in the dish and in rodents and found that it looked like it might be protective. And that led to this trial of inosine, which is a natural chemical in our bodies that our bodies turn into uric acid. So it's basically a way of increasing the uric acid levels and seeing whether that in turn will slow the decline of Parkinson's disease.
Terrific. Well, again, I think this list is incredibly promising. It shows us that not only do we have a lot of possibilities, but many of those possibilities are now fairly far advanced in the clinical trial process, and we'll know something more by the time we do this show a year from now. Let's move on and start talking about what progress is being made in terms of symptomatic management. Again, the difference we're talking about here is that the first group are all strategies that would actually do something to slow down the progress of the disease. Now we want to focus a little bit in the meantime. What are we doing to just help us all live with Parkinson's in a good way, in a day-to-day basis, by managing our symptoms better? And these are two developments that took place in 2015 to manage some of the motor symptoms more effectively of Parkinson's. And they both, Mark Fraser, have to do with delivering Carbidopa, Levodopa, the standard drug in Parkinson's, in a more continuous way, one by way of an extended release capsule, the other by way of a gel, which would actually be pumped directly into the small intestine. What are the advantages of this over the standard pill form of taking Carbidopa, Levodopa?
Sure. Well, and Carbidopa, Levodopa has been around for a long time and is very effective in treating many of the motor symptoms of Parkinson's disease. While it's very effective, it does have some challenges. For example, over time, if you take it as a pill, the levels of Carbidopa and Levodopa in the brain vary over time, and so you have these peaks and troughs of the levels of Levodopa that really can cause disruptions in alleviating the motor symptoms. Also, over time with extended use of Levodopa, it can start wearing off and not working because we have what we call these off episodes. And so because of this these peaks and valleys that occur with taking the pill form or the traditional form of Levodopa, the goal of these approaches was really to provide more of this continuous continuous stream of Levodopa to the brain, so you would reduce this wearing off effect. So it's really putting more of a cruise control on the amount of drug that's in the brain and really just providing benefit over time so that these peaks and valleys and peaks and troughs don't occur.
And both these now are approved for usage here in the United States. They've actually been approved elsewhere in the world prior to their approval here by the FDA. We also see a note at the bottom of the screen that says that a Levodopa inhaler is in late stage testing. It's, as I understand it, was thought of as kind of a rescue drug, that if you're kind of in between pills and you need something to carry you over the hump, it would provide sort of instantaneous relief. Is that right?
That's right, and you know that is a need in with patients to have, when they feel like there's a sudden off occurring, to have a quick acting drug that can rescue them from the drug wearing off is really important. And all of these approaches, you know, as is indicated, are using the same drug, but really just engineering different ways to deliver them that could have a benefit on the quality of life.
All right, so some good developments this year in terms of better symptom management when it comes to motor effects. But let's turn now to what an additional question, which we've had a couple of questions come over the course of our hour so far that we want to turn to next, which is also having to do with motor symptoms and better ways, some new ways of providing surgical assistance to living with Parkinson's. We're all familiar with deep brain stimulation, which Tim Greenemeyer is especially effective at dealing with tremor, not something that slows progression of the disease, but very effective in terms of symptom management of tremor in particular. Now there are a couple of new twists on this, which I think are interesting. This first one, Tim says, talks about a kind of smart form, I guess, of DBS, where the stimulation would only be triggered when you need it, which sounds intriguing and has obvious benefits. Can you describe where we are with that idea?
Yeah, let me first describe how conventional deep brain stimulation works. The electrode is placed in a certain area of the brain, and it is essentially like a pacemaker. The controller for the pacemaker is outside of the brain. It's in a little pack. It's under the skin of the chest typically, and your neurologist will typically adjust that after surgery and then as needed every few months. And basically it's stimulating continuously at a certain frequency and a certain voltage and so on. And it stays that way until your next visit for readjustment. The idea behind what we call closed loop deep brain stimulation is to use some sort of signal from the patient that informs the stimulator that now I need the stimulation or now I don't need the stimulation because I'm doing well. So there are a number of labs around the world and companies around the world that are working on this, trying to figure out, number one, what's the appropriate signal? Should it be something external, such as your tremor, or should it be entirely internal, measuring brain activity that might be associated with tremor or gait, and then doing the signal modification to make that adjust the stimulation of the DBS electrodes according to how you're doing. And that has several advantages. Number one, you only get the stimulation when you need it. It can be quiet most of the time if you're doing all right. Secondly, it's going to prolong battery life, and replacement of the batteries can be a big hassle and involve minor surgery currently. So I think it has a great deal of promise, but I don't think it's immediately on the horizon yet.
We have an interesting question from William, which is, how do you identify which cells are miscommunicating to target what you target with DBS? Is it looking for cells that are miscommunicating? Is it electronic signals? What is it? Or is it just different areas of the brain, Tim, that we're trying to identify when we figure out where to place these electrodes?
Yeah, that's a great question. It's circuits if you have ever seen a circuit diagram of a piece of electronics, you know, this transistor is connected to that and makes this turn on and so on, you have the equivalent of circuits in the brain where you have some nerve pathways that turn on other nerve pathways, and you have other neural pathways that turn them off, and there's inhibitory or excitatory. And people have mapped these out using primarily animal models of Parkinson's disease, and have figured out that in Parkinson's disease, in particular, there are certain areas that have abnormal activity. Either they're firing too much, or they're firing in a pattern that is wrong and that is leading to the symptoms of Parkinson's. And it's been found that if you interrupt that signaling, either by DBS or by actually killing those cells in some cases, you can have great effects on the symptoms, including tremor, and dyskinesias are also very effectively treated with DBS and related treatments. So DBS is stimulation of these cells that sort of changes the way they fire such that the symptoms are no longer as apparent. The next topic that you're going to bring up, Dave, the focused ultrasound, instead kills those cells and also has effects on the symptoms, but from a different approach.
Well, just stay with that if you would, Tim, and explain what it is. This is kind of the idea is you get the benefit of surgery without surgery because there's no incision. But the effect is kind of like the old-fashioned pallidotomies that took place, where we kind of burned a part of the brain. Is that right?
That's right. In many cases, the areas that you can stimulate with a DBS electrode, if you simply destroy those areas in a very precise manner that used to be burned, as you said, with pallidotomy or thalamotomy, you get many of the same benefits. With the focused ultrasound, basically you're focusing the energy from the ultrasound machine in a very precise manner right through the skull, and targeting these areas that are that have abnormal activity, killing them, and this is resulting in benefit in terms of primarily tremor, but also probably dyskinesias for many people. There's an equivalent technique that's been around for a while called gamma knife surgery, which instead of using focused ultrasound uses focused radiation to do the same thing, so it doesn't require surgery and achieves the same goal. So for those people who don't really want to have a surgery, or for medical reasons can't have a surgery, this is a reasonable option. It doesn't require the surgical incision. There's no batteries. There's nothing placed in the brain that might get infected, and so on. But the downside of this and gamma knife is that it's permanent. It's not reversible. You can't adjust it like we do with DBS. You know, if the lesion or the damage isn't in exactly the right area, we're stuck with the result that we get. It has pros and cons like all other treatments.
And Tim, is it still being tested, or is it now available?
As far as I know, it is only in trials at this point.
And one other question comes in from Sigrid, which is, if focused ultrasound does become more available, could one person have both focused ultrasound and standard DBS? Not on the same side of the brain, because they deal with the same side, or the same area of the brain. So in one case, you're destroying that area. In the other case, you're stimulating it. But if you've destroyed it, there's nothing to stimulate. And if you've got a stimulator in there, you can't destroy it. So I think, at least on the same side of the brain, they're sort of mutually exclusive. Okay. let's move on and talk some about where we are with a really key area, which is the non-motor symptoms of the brain. And I'm not getting the screen on my screen, so there we go. Let's talk about this some, because as many of us know, the non-motor symptoms of Parkinson's can be as problematic, or if not more problematic, than the motor symptoms. And Dan Cannell, let me bring you back into our conversation at this point, because I know you've said when we've talked on other occasions that for you, fatigue in particular has been problematic. So maybe one way to get at this aspect of where we are with accomplishing more progress with treating these effects is to have you tell us sort of what the problem is, and then we'll talk about where we are in terms of trying to find a solution.
Well, for me, the problem is that that sort of in the middle of the day, I'll just get really tired and just want to fall asleep. And one of the ways that I combat that is through the regular exercise. It seems it's counterintuitive, but the more I exercise, the less tired I am. But it's a problem that's been ongoing that it just takes more energy to sort of do the day-to-day stuff that encompasses living.
And have you participated in either trials that have to do with some of these non-motor problems, Dan, or exercise trials to try to identify ways to combat them?
I haven't participated in any exercise trials, but sort of following in the footsteps of some recent publicity, a friend of mine who also has PD and I recently started taking boxing lessons, which has been tremendous fun and a great way to get out aggressions, and has been we've noticed improvement in our coordination and balance. So I think that anything that you do to get out there and keep your body moving is probably generally healthy for anybody is probably good for Parkinson's.
Right, and well, I'm not sure if you're participating in the so-called rock-steady boxing approach that's been developed specifically for Parkinson's. I know a lot of people are excited about that. Let's talk more about this. In some ways, it seems kind of like a good news, bad news problem, Mark Frazier, that the good news is, as Dan suggests, that exercise really appears to be one of the things that we can do that's the most effective in combating some of the symptoms of Parkinson's, both motor and non-motor. So there, on the one hand, it's sort of frustrating. The bad news is that we haven't made more progress in developing other kinds of pharmaceutical therapies. The good news, I guess, is this is something where we can really make a difference ourselves, right?
Yeah, I mean, this is a big area of research, and I think increasingly being appreciated in the research community, that there are opportunities to really accelerate progress and get some real, just not just understanding, but new treatments for the non-motor symptoms of Parkinson's. And I think patients and treating neurologists have probably appreciated this for a while, that these are major issues, but the research community just recently, I would say, is catching up and focusing a lot of attention on these different symptoms. And I think one of the challenges to this is because every patient experiences a different constellation of these non-motor symptoms, it's really hard to understand from patient to patient what's causing those symptoms, whether it's due to Parkinson's disease or there are other related factors. And because it's so variable from patient to patient, to develop some general understanding about the biology and what's happening in the brain and the body as it relates to Parkinson's and what's causing these non-motor symptoms. So I think the good news is that there's a lot of effort and attention now more recently, and a lot of funding dollars being put towards these non-motor symptoms. I would say it's a little bit less well understood than some of the traditional motor symptoms of Parkinson's, like tremor and slowness and rigidity. And so that, I think, is causing some delays or challenges in getting new treatments for these non-motor symptoms.
And Tim Greenemeyer, can you comment on the first point, which is a drug for Parkinson's psychosis. And we have a question coming in which says, at what stage does psychosis begin, and what are the signs? So maybe, Tim, if you could describe some of what Parkinson's psychosis actually is. Is it part of the disease? Is it part of the drugs we take? What exactly is it, and what does this new drug do that would help deal with that symptom?
Sure. Psychosis is common but not inevitable in Parkinson's disease. So some people never develop it, and other people develop it relatively early. It's caused by both the disease and the effects of Parkinson's disease on the brain, and it's made worse or even caused in some cases by the medications that we need to use to treat the Parkinson's disease. And it consists of things like hallucinations, but hallucinations alone, the typical hallucinations that many people get of just seeing little furry animals or little people that are not particularly bothersome, I wouldn't personally classify as hallucination or as psychosis, and may not need any treatment if they're not bothersome to that person. But if the hallucinations become frightening and begin to affect that person's behavior or their interactions with others, then it's a problem. People may become delusional and feel that people are after them, so they become paranoid, or they believe that their spouse is having an affair. I've seen that fairly commonly. So it's this constellation of paranoia, changes in behavior, hallucinations, and so on that we call psychosis. Now, we've traditionally had a very difficult time treating this. You can reduce the psychosis if you reduce the Parkinson's medications that we use to treat the motor symptoms. If we reduce the dopamine agonists, things like the Mirapex and ropinirole, or we reduce the Levodopa, we can reduce the psychosis. But of course, then the Parkinson's motor symptoms get much worse. On the other hand, we can use typical antipsychotic drugs that are used for schizophrenic patients, for example. And these drugs block the dopamine system, so they can be very effective in reducing the psychosis, but they will also make the Parkinson's symptoms much worse in terms of at least the motor symptoms. So we've traditionally just been in a double bind. We can't really effectively treat the psychosis without making the motor symptoms worse. This new drug, Nefluzid, is a new form of an antipsychotic medication that reduces the psychosis without having a significant effect on the motor symptoms of Parkinson's disease. So it's a real step forward for those people who do have this psychosis.
Well, it sounds like a significant improvement, and as Mark suggests, it also sounds like, and I think this is good news, that the research community is going to continue to focus on these non-motor symptoms, cognitive issues perhaps in particular, are ones of great worry and concern to the patient community, and it sounds like hopefully there will be that much more emphasis on that in the research world in the year ahead. I'm going to move us on to our next point, which has to do with the way in which patients and researchers can connect better. Don't want to spend lots of time on this as we want to get to more of your questions, but Dan Cannell, I want to bring you back to have you comment on this. I know this is something that you've been interested in particular, not only in participating in clinical trials, but also in participating in some of these online applications or smartphone applications that allow researchers to collect more and more data. Describe what you've done and why that's of interest and importance, do you think?
Sure. I've done a couple of what are I guess wearable or online studies. I participated last year in a couple of telemedicine studies, and they're all, I think, designed to help researchers and help clinicians get better access to patients to improve outcomes and quality of care, which is important for everybody with Parkinson's. As far as ways to get involved, as we mentioned, there's the Fox Trial Finder, but I think that most people who go to a neurologist who's a movement disorder specialist specialist at a major medical institution would be aware of, their doctor might be aware of different studies and opportunities to participate and get involved as well. So the more information we get to the researchers, the more ammunition they'll have to improve outcomes and quality of care, and then hopefully find a disease-modifying therapy.
And as we move to our next point on this, Mark Frazier, maybe you can just tell us what big data gives us. I mean, why is it important for you at the Fox Foundation to collect information from not just Dan, but thousands and thousands of people? What does that give us, I guess, that that's different from 100 people participating in a clinical trial?
Yeah, and I think it's a good question. The traditional way of doing research is having people participate in a clinical study or clinical trial. They come into the research center and participate and fill out forms or undergo certain tests. And that's somewhat limited in scope in terms of the amount of data that can be collected, because those studies are expensive and often limited to a certain number of patient volunteers. What technology and the changes and revolution in technology has brought us is the opportunity to acquire lots and lots of data on thousands of people. Some of it may be more useful than others, and this is the experiment, or one of the experiments of Fox Insight, is really to see if we can acquire lots of data on lots of people, and then use some large data analytic techniques that are becoming more and more popular and improved every day to really detect patterns in Parkinson's disease or Parkinson's progression, potentially identify subtypes of certain people that progress in one way and others that progress in a different way. And using these large data sets and data from lots of people would be the way to do that, whereas the traditional way of research and really following individuals on a smaller scale really would not lead us to those insights. And having that possibility of large data sets and the analytics on top of that, I think, is really exciting for Parkinson's.
This is Dan. Bringing it back down to the patient level, being involved in studies that generate data also allow me as a patient to see more information about my symptoms and condition, and help me at least better manage and interact with my treating physicians about what medications and fine tuning was best for me. So in addition to providing lots of important data to the researchers, it provided me a personal benefit as well, which I think was a pleasant side effect of participating in the data-related studies.
It's a great point, and as we move to this next slide about what we can all do today and participating in research, participating whether that's in Fox Insight or Fox Trial Finder or both, I think the point you're making, Dan, is that there's a kind of enlightened self-interest to participating in research. It's not just doing good for others, you're doing good for yourself, because you're getting that much more information. And I know you said that's how you actually found your neurologist, because you by participating. Yeah.
Absolutely. I participated.
That's great. Let's get to our questions in our remaining time, as we allow people to also take note of this and the ways that they can get involved. A question coming in, Tim, about what we really know at this point about exercise. We see the bullet point there that there's no best exercise. It's really the exercise that you'll do on an ongoing basis that's key. But summarize for us what we now know definitively about the benefits of exercise for someone with Parkinson's.
Well, I think we know that exercise clearly has benefits. As Dan was saying, just exercising on a regular basis, you feel better, your fatigue is less. And I have many patients who, if they can't or don't exercise on a given day, just don't feel as well and don't do as well. But perhaps more importantly, there's increasing evidence that exercise on a regular basis may change the progression of the disease. It might actually protect your brain and the studies are ongoing to sort of sort this out at this point, but that's the hope that, just like we talked about all of the therapies aimed at synuclein to try and slow down the disease, exercise may be a non-drug therapy that will have the same effect, at least in part. So I think that there's great hope in this. As we've said, there's no one right exercise. So what I typically recommend to people is a combination of cardio/aerobic and strength or resistance training, because both have shown benefit and may actually change or slow the progression of the disease.
Get to more of our questions. And a number of people have raised questions about some things that we haven't discussed yet. Here's the first one about glutathione. Tim, why don't you take that one as well? We talked about briefly that the other day on the phone. What is glutathione? Is it something that has possibilities?
Sure. Glutathione is a natural antioxidant compound, so it protects against the oxidative stress and damage in our cells. All of our cells make it, and it's beneficial. And it's been known for probably 20, 25 years that glutathione levels in the brain, particularly in the substantia nigra, which is most affected in Parkinson's disease, are reduced. So you have less glutathione, you have more oxidative damage. And it makes sense then to try and increase the glutathione levels in the brain. The problem is that we don't know how well glutathione actually gets into the brain. It doesn't look like it gets in very well. There are no good controlled studies that suggest that it works. So I can tell you, and many of you may know, that there are practitioners out there who will offer IV glutathione to people, saying that it's a wonder drug, and it's clearly not a wonder drug. Whether it does anything at all is unclear at this point. There are some ongoing trials of glutathione, and another compound that can be converted into glutathione that I think probably has more promise, called N-acetylcysteine, or NAC, for the same purpose. But right now, there's not enough information. I certainly don't think that charging people for intravenous infusions is warranted at this point.
I'm really glad you mentioned that. I think there are marketing attempts out there to take advantage of things that are ideas in theory at this point for which there is not adequate clinical evidence, and I think we all have to be wary of those sorts of marketing approaches. Question coming in for you, Dan, about fatigue in particular. This person writes in, it's really difficult to initiate exercise when you have fatigue. So how do you overcome that?
I think that for me, I start small and keep building. Do what you can and as much as you can in any reasonable day, and over time your endurance and stamina goes up. I think the key to any exercise is not to overdo it. Do as much as you can and build stamina over time, rather than do it all at once and get hurt. I think that the more movement that we do, that we engage in, the better off we are.
Question, Mark Frazier, on stem cells. Has that been abandoned as a therapy? This is something we've talked about before, that once upon a time, not really once upon a time, even 10 years ago, that would have been at the forefront of this discussion. It seems less now. But where are we with stem cells? I know that's a big question to make you answer in a minute, but do your best.
Sure. No, and I wouldn't characterize it that the field has abandoned stem cells. I think based on earlier work, probably in earlier 2000s, there has been an appreciation that getting a stem cell to integrate into the complex circuitry that is a human brain, if you just stick something in, is challenging. And so there is a lot of work ongoing to understand how to integrate a stem cell into the brain and what's needed. There's also been some really remarkable discoveries where we've been able to take a skin cell from a human and create a stem cell in a dish. And these types of experiments have been used in research and drug development programs to really create what we call a disease in a dish. And they have been, while not a therapy per se, they have been important models of Parkinson's disease to really test new therapies in a dish. So there's still utility there. And then the final point that I would make is going back to the therapy aspect. If we are able to figure out how to integrate a cell into an existing circuitry, an existing brain, most likely the stem cells would treat the dopamine-related symptoms of Parkinson's disease. So that is to say, most of the movement, motor symptoms of Parkinson's. But as we discussed, there's this growing appreciation of the non-motor symptoms of Parkinson's disease that a stem cell that's developed into a dopamine cell may not address. And so simply putting a stem cell in may not solve all of the issues related to Parkinson's. But I think it still warrants research. I still think it has promise. It may just be a tougher road to hoe than we originally thought.
Another tough road. Yeah, go ahead, Tim.
I just wanted to add, relevant to the glutathione discussion, people will find online that there are clinics in various countries or islands outside of the United States that are offering stem cell therapies and guaranteeing cures, and it's all snake oil. So it's not ready for prime time, and it should not be considered as a therapy at this point.
Yeah, thank you for adding that. Another difficult problem that we have just touched on, and Tim Greenemeyer picked up on this too, which is the question about where are we with treating the mood disorders, problems like anxiety, aggression, and depression that are also part of Parkinson's disease.
Well, it's as Mark was saying, it's difficult. We don't understand fully the biological basis for these symptoms in Parkinson's disease. So right now we are essentially stuck with drugs that we use for other purposes. For depression, we use typical antidepressants. For anxiety, we use anxiety drugs that are used for the general population with anxiety. And we use them to varying success. Sometimes they work, sometimes they don't. But there's a great deal of interest in trying to develop better therapies, more specifically targeted to those with Parkinson's disease, because these symptoms arise from different causes in Parkinson's versus the general population, but we're not there yet.
And a question from Paul, which has to do with whether or not how your Parkinson's originated affects your treatment. Paul wants to know if your Parkinson's is caused by a genetic mutation versus potentially an environmental exposure, and that's still, of course, a quest in Parkinson's research, is identifying the causal agents. But does that causal agent, Tim Greenemeyer, make a difference in how your Parkinson's is treated?
Right now, no. But the hope is that we can develop what is often called personalized medicine for Parkinson's disease, so that if you have a specific kind of mutation that it has caused your disease, we have a specific medicine to treat it. And there's a great deal of interest in this. So probably the example that's closest to fruition is a form of Parkinson's disease caused by mutations in a gene called LRRK2. And there's a great deal of interest in developing drugs that specifically impact LRRK2 and may be beneficial. And this is probably a relatively large population of people with Parkinson's disease. So that's the goal, but right now it doesn't make any difference. And for most people, excluding those with clear genetic causes of the disease, we don't know what has caused any individual's disease. Although we may be suspicious in some cases, we don't know with any certainty.
Let's move to some last comments as our hour is coming to a close. And again, here's how you can find out more about participating in research by going to foxtrialfinder.org, which Dan and others have mentioned throughout the hour. I said at the beginning that part of what's exciting to me as someone with Parkinson's is that we're no longer talking about just a wish list. We're talking about a work in progress list. And that, I think, is extraordinarily exciting. But as we look forward to the year ahead and beyond, I'd like to hear from each of you about what your hopes are about the kind of progress that we will continue to make. Mark, a brief comment from you first.
Well, I guess I'm certainly energized by that list that you had put up, and I think the field is really energized about the progress that's been made and the trials in human testing already. I'm hopeful for more trials to enter human testing. I'm optimistic that many of the research and the programs that we've supported will enter human trials in 2016. I also think, and I'm hopeful, that we are going to have better biomarkers that will be used to inform these clinical trials. So we would benefit, and it would transform the field to have an imaging agent to image alpha-synuclein in the brain in human beings. And we've supported a lot of research in this regard, and I'm optimistic that we're going to have an agent in humans next year that will be able to be utilized in many of these clinical trials to see if some of these synuclein-based approaches are actually breaking up the clumps in Parkinson's disease, the clumps of synuclein.
And Tim Greenemeyer.
Well, Mark has stolen most of what I wanted to say, but what I would actually say is there's no good time to have Parkinson's disease, but this is the best time that there has ever been to have Parkinson's disease, because I think we are close to having disease-modifying therapies. I tell patients that I see that I think in the 3 to 5 year range, we will have something that won't be perfect, but something that will impact the progression of the disease in a very positive way. And I'm really excited about this. I think we are getting closer and closer with all of the list of approaches that we have already discussed, but other approaches as well. So I am really optimistic at this point.
And Dan Cannell, you've been on literally a safari, and you're on a kind of ongoing research safari as someone who participates in so much clinical trial experiences. Your hopes for the year ahead?
Well, I look at that slide with the different therapies in development that have potential disease-modifying effects, and I think of them all as shots on goal, and I hope that one of them makes it past the goalie and scores a point.
Great way to end. Thank you all so much, Dan Cannell, Tim Greenemeyer, Mark Frazier, for participating in this session. It's been, I think, really useful, and I think it energizes us as we go forward into 2016. We'll be sending all of you a link to the webinar so that you can listen again or share it with others. There will also be a survey link in that email so that you can let us know what you thought about today's presentation. We also hope that you'll mark your calendar for our first third Thursday webinar of 2016. That will be on January 21st at 12 Eastern, when we'll talk about dyskinesia and the research progress that's being made in treating that particular problem. Finally, again, if you're on Twitter, you can join us tomorrow at 12 Eastern, 11 Central, 10 Mountain, and 9 Pacific, where we'll continue this conversation on Twitter. The hashtag is FoxPDChat. So thanks all very much for participating in our webinar today, and all of us at the Fox Foundation wish you the happiest of holiday season. I'm Dave Iverson.